Welcome to Sci-Chat Mindset. Today we're diving into the fascinating world of DNA, deoxyribonucleic acid, the blueprint of life. DNA is a long, double-stranded molecule found in every cell of your body, carrying the instructions for growth, development, and functioning. It's tightly packed into chromosomes inside the nucleus. But how do we actually extract this vital molecule from a cell? Let's find out. Each strand of DNA is composed of three key structural components: a phosphate group, a ribosugar, and nitrogenous bases. These bases are divided into two types, which include adenine and guanine as purines, and cytosine and thymine as pyrimidines. In the DNA double helix, adenine pairs with thymine through 2 hydrogen bonds, while guanine pairs with cytosine through 3 hydrogen bonds, ensuring the stability of the structure. Extracting DNA is a fundamental technique in molecular biology, essential for experiments like genetic testing, forensic analysis, and research. One of the most reliable methods for extracting DNA is the Phenol-Chloroform extraction. This method uses organic solvents to separate DNA from proteins and other cellular components, yielding pure DNA for further analysis. Let's break it down step by step. Step 1. Cell lysis, and begin by breaking open the cells to release DNA. This is typically done using a lysis buffer 10-set lysozyme containing detergent to disrupt the cell membrane. Step 2. Add an equal volume of phenol-chloroform to the lysate. Phenol denatures proteins, while chloroform helps separate the aqueous and organic layers. Centrifuge the mixture, separates it into 3 layers. The upper aqueous layer containing DNA, and lower organic layer with phenol proteins. Step 4. Carefully transfer the aqueous layer to a new tube, being cautious not to disturb the protein interface. Precipitate the DNA by adding cold ethanol or isopropanol. DNA is insoluble in alcohol, causing it to form visible strands. Wash the DNA pellet with ethanol to remove impurities. Then finally resuspend it in a suitable buffer for storage or downstream applications. And that's it, you've successfully extracted DNA using the Phenol-Chloroform method. If you found this tutorial helpful, don't forget to like, share, and subscribe to Sci-Chat Mindset for more exciting science tutorials. Thank you for watching.